other thing is we're going to talk about the microbiome, all those bacteria that you have in you, and that's also another good topic to talk about after you've eaten, and the bacteria are doing really well. So the microbiome is an ecosystem that you have in your body. Sometimes it's called your second genome because all the bacterial genomes provide other genetic functions. Sometimes it's referred to as another organ. It's like you discovered the heart or something like that because it has such an important role in human physiology. And for those of you who don't know more precisely what it is, the definition of the microbiome are the communities of microorganisms that share your body with you. And you'll hear lots about these microorganisms, and it's an ecological phenomenon. And then you'll also hear the term metagenome. So if you think of all those microorganisms, each one of which has its own genome, the collection of all of those genomes is your metagenome. It's like you have a second genome because of all of that genetic material that you carry around with you. So you're actually mostly bacterial cells. If you counted all the cells in your body, 90% of them would be bacteria. So you're all just big bacterial communities with a few human cells sprinkled in there. And if you think about how many bacteria you have in you, it's an astronomical number. Sometimes they say stars in the sky or grains of sand on the beach or something like that. In your mouth, you have more bacteria than there are people on the earth. So we're talking about vast numbers of organisms. And since we were talking about the mouth, here's what the community of microorganisms in saliva looks like. There we go. Here's what the community of saliva is. These are all different taxa of bacteria. If the font is large, it means it's an abundant organism. And you see many of these have very small fonts. They're not very abundant. But because they're not abundant doesn't mean that they're unimportant, because even organisms that are present in low amounts can still be making molecules or having a big impact on the host. So the goal is to try to, in different tissues, describe what these microbial communities are. And that's what studying the microbiome is really all about. One other concept is that if you do arithmetic and figure out how many microbial genes you have compared to human genes, you have way more microbial genes than you have. So right now, after lunch, as all of those nutrients are being catabolized by both your body and by your microbes, 99% or more of the gene activity going on in your body is from your bacteria. And the host genetic activity is significant, but it's dwarfed by all the microbes that are there. Why do we have them? Well, they provide lots of functions that we don't have to code into our genome. They can synthesize molecules that we don't have to synthesize. They can degrade molecules of all the different types of nutrients we have, so we don't have to code that into our system. They protect us against infection, partly because there's just so many of them that when a pathogen tries to infect you, it's hard for that pathogen to get in. They do many, many things. And that's been selected for by evolution. So they're very, very tightly integrated into our system. So here's the basic microbiome analysis that I'm going to be talking about. The question is, if you take a sample, saliva, stool sample, nasal sample, skin sample, whatever it is, you just want to know what is in it. And the key concept is that we just extract all the nucleic acids in the sample, we sequence everything that's there, and then there's various kinds of analyses we can do. If we want to know what the organisms are that are there, taxonomic analysis, We might, for bacteria, we might target just sequencing the 16S ribosomal RNA gene. That's a gene that is used to define different bacterial taxa. And we end up with a table like this. So from one person who gave us a sample, this is the table, this is the data point that we would get. It lists all the different taxa and their relative abundance based on how many sequences we saw. But we could also just ignore what the organisms are and do comparisons to databases and ask what genes do we see and what's the relative abundance of those genes, because from a real ecological point of view, you could think of a community as not different organisms, but just as different functions, different genes that are present. And the other way to look at that would be to look at metabolic pathways. So again, you take all the sequences, compare them to a database, and you would now have the different pathways of metabolism and their relative abundance based on how many sequences we have there. So these are three different ways of looking at the microbiome, and we use all of these. So here's an example. We took samples from 6 different body sites: the posterior fornix of the vagina, a stool sample for the gut, anterior nares, that's your nostrils, tongue dorsum, your tongue, buccal mucosa, your cheek, and the supragingival plaque, your tooth. And so we took samples from all of those, and we extracted all the nucleic acids, did sequencing, made a table for each one of those samples. These are from a couple of hundred different people. And then we compared those tables. You know, sometimes you might just say, if all the organisms present in one table are present in the other table, and the ones that are absent in one table are absent in the other table, they're identical. Or it might be that if an organism is present in one table but not in another, now they're a little different. And so we try to figure out how similar or how different the tables are, and then we use that to build this tree. This is a tree that it's kind of bent around so it'll fit onto the slide. And each one of these lines is a different sample, and so if 2 lines are right next to each other, it means their tables are very, very similar. And if 2 lines are very far apart from each other, say here and here, that means that their tables were very different. And look at what happens when we do that. All of the samples from each of the body sites cluster together. In other words, all the stool samples are much more similar to the other stool samples than they are to the samples from the other parts of the body. In other words, each of those 6 different body sites has a microbial community that is characteristic of that particular body site. And if you look, say, just within the stool samples, you can see there's one group here, there's another group here, and there's a third group here. That means that of all the people in this study, you fall into 3 different classes of what kind of community you have in your gut. And this has been seen many, many times. Some people have a bacterial community in their intestine that is dominated by a taxon known as Bacteroides. Some have one that's dominated by Prevotella, and some have one that's dominated by gram-positive bacteria, something like that. So you're not only all full of bacteria, but you're either a Prevotella person or a Ruminococcus person or a Firmicute person. And you can see this also. Here's the vagina. You can see there's 2 groups here. This is lactobacillus, so these are women who have their vaginal microbiome is dominated by lactobacillus, and these women do not, and so forth for the other communities. So that's the awesome power of microbiome analysis. We can just take these samples, extract the nucleic acids, sequence them, make a tree, and we get biological information out of it. We see how things cluster together, and we can see what organisms are more common in one tissue than in another tissue. And if you do this in a sick person and a healthy person, or person on one diet or another diet, or all these different conditions, you can also separate people according to their microbiome that way. And the other thing that's interesting is the tongue, the cheek, and the tooth are all in contact with each other, and yet their communities are distinguishable from each other, and that tells you that the microbial communities are very tightly bound to the tissues where they are. They interact with those particular host molecules, that environment in terms of the nutrients or whatever else is there. Whether it's immune privileged or whether it's sensitive, things like that. All of those select for a particular type of microbial community. And that played out throughout your whole body. So let's not forget about viruses. When we do the sequencing, we can look for viruses too. And so here's 100 healthy people, and this is the number of different genera of viruses we find. And on average, a typical healthy person is infected with 4 or 5 different viruses. So you're not only full of bacteria, you're full of viruses too. And here's some people that have 15 different genera of viruses. These are lots of different viral taxa. These are the 100 people that we use. You can see some of those. Here's a virus. This is a herpes virus. Almost everybody has this. Then there's other viruses that only 1 or 2 people have. So you also have your own communities of viruses. They don't fall into as nice classification as those 3 types of intestinal people. The viruses seem to be much more individualized, much harder to group. But nevertheless, it's part of your microbiome, and these viruses are presumably doing something in the body. We don't know very much about what it is. Okay, so how will the microbiome affect medicine? And, there's 3 general ideas in general: maintaining health and preventing disease, curing disease and restoring health, or diagnosing disease. So prevention, therapy, and diagnosis. Those are the 3 biggies. So let's start with how do we maintain health? What do we do? So the obvious thing is it may have to do with diet. So what do we know about how the microbiome affects diet? So it's hard to do dietary studies on humans because after a couple of weeks they want to eat other things than what they're supposed to eat on the project. But with monkeys, we can put them on diets. And so here's 2 diets that these monkeys were put on. Monkey chow, which is kind of a soy-based diet, and then TAD, the typical American diet. Lots of calories from fat and things like that. The first time this experiment was done, the monkeys took the food from the typical American diet and threw it back at their keepers. They wouldn't eat it. So we had to change what the formulation of that diet was. But anyway, what you can see is, again, these are all different monkeys on different diets, and we do that thing about comparing their tables after we sequence, and this is all from poop. And you can see that the monkeys on the chow diet all cluster together. Their microbiomes are all pretty similar. And here's one group of monkeys that were on the typical American diet. And then here's another group that's sort of a mixture of the 2 diets. But nevertheless, it's obvious that diet can have a big effect on the microbiome. So if you wanted to maintain health, you could presumably manipulate your microbiome by your diet and kind of control what's going on there. You could go out one night and eat a lot of things that are bad for you and drink a lot, and then the next few days you could manipulate your microbiome by eating things that would put the right organisms back in place. But this doesn't really answer the question is, what's a good microbiome? What do we want to shoot for? So somebody wrote a book. There's already a book about a microbiome diet. It's not a very good book. I don't recommend it, but we're going to have a lot of books about the microbiome diet. We need to learn how foods affect it. We need to figure out how to adjust things, maybe probiotics and other things. So this is sort of the game, is to understand how these different things affect the your microbiome, so you can manipulate it yourself. So to figure out what's the golden microbiome, the thing that we all should try to have, we have a project going on where we look at the microbiomes of athletes. So these are professional athletes. They race their bikes. They go down mountain bike racing every week. They're very fit, very disciplined, very motivated people, all wonderful habits if we all had those. So if we can just get the right microbiome, we'll become fit and disciplined and motivated and have all of these good characteristics. So so far we've actually looked at more than 23, but that's the idea. And so once again, we got stool samples, and also from healthy people who are not athletes, and also from people who compete with these athletes in the races, but they never win. They're sort of like weekend warriors. They're out there riding their bikes, but they're not professional athletes. And so the athletes are in the green here, and the other people with the Human Microbiome Project people are in the red. And so again, we compared their tables and we get this tree. These colors here are the different organisms that we find in their gut. But as you can see, here's one cluster of the tree. Here's a bunch of athletes that have microbiomes that are all similar to each other, and they're different from just the healthy people who are not athletes. They cluster together here, and then there's some smaller groups here like there was in the monkeys where you have a mixture of athletes and healthy people. Maybe some of those healthy people were actually athletes and we didn't know it. But there's clearly something about their microbiome that's different, and we want to analyze that and try to figure out what it is, because that's what we would like to all have. So one of the things we find is they're enriched for Prevotella. Remember when we looked at the stool samples in that round thing, there was the Ruminococcus people and the Prevotella people and the Bacteroides people. So Prevotella is one of these groups. And then there's another bacterium here, Methanobrevibacter smithii, which is an archaebacteria. It's different than the bacteria that we commonly deal with. There are three kingdoms of life, as you may know. There's the eubacteria, there's the archaebacteria, and then there's the eukaryotes. So this is a bacteria from that other kingdom of life, and it's highly enriched in the athletes. So why might that be? So the archaebacteria, in particular, M. smithii, it's a methanogen producer, and it produces methane by starting with really elemental things, hydrogen gas, CO2, acetate, formate, methanol, things like that. It can use that to grow, to make all the molecules it needs and derive energy from that. And of course, these are the end products of metabolism of all the other bacteria that are in the gut. So what this bacteria is doing in your gut is the food comes in, the other bacteria metabolize it, and then M. smithii takes the end products from those and gets more energy and nutrients out of the food than they could get out of it. Plus it kind of cleans up the gut, and the other ones can now grow better and do more metabolism anyway. So that's the theory, that having this organism there in the community gets more out of everybody. So we have to prove that yet, but that's an interesting observation. Okay, let's take a look at therapies, curing disease and restoring health. So let's take the example of Clostridium difficile-associated diarrhea. Some of you may be familiar, and some not. I made a very simple picture here of what people think goes on. Here's a normal microbiome, has all these different organisms in it, and these dark ones are Clostridium difficile, which is a bacteria that is resistant to many antibiotics, and it also produces toxins, which can do damage and cause diarrhea. But normally, you can carry some of it, and it's kept at a very low level by all those other bacteria that you have in your gut. Now, occasionally, people are treated with antibiotics because they go into the hospital, let's say for getting their appendix out or having some simple surgery or some other procedure, and they're put on antibiotics to protect them against getting infected just for the time they're in there. And when that happens, you destroy this microbiome, so most of the bacteria go away, but Clostridium difficile, which is resistant to the antibiotics, remains, and it can now expand. It can become more abundant. And when that happens, you can get diarrhea and this disease. And how do you get rid of it? Because you can't treat it with antibiotics because it's already antibiotic resistant. So people who have this condition are, it's a horrible situation to have, and it's very difficult to treat. So it turns out that if you take fecal material, the gut, from a healthy person, and you introduce it into this Clostridium difficile intestine, now there's suddenly a lot of good bacteria around, and they colonize again, and they reduce the amount of C. difficile, And the doctors that I've talked to who've done, 60 or more of these kind of transplants say that it's amazing. Almost all of them work, and you see the effect in 3 days. So what other medical treatments can you think of that in a few days a terrible condition is gone and it works in almost everybody? So anyway, that's sort of one hot area of microbiome therapy right now. And of course, people are trying to figure out artificial microbiomes so you don't actually have to treat somebody with the stool sample from a healthy person. You can just put in a bunch of bacteria and do the same thing. Here's another one that's not quite as gross. This was an experiment we did. We're looking at acne, right, a skin condition, and we look at bacteria from healthy skin or acne skin. And these are all members of the species Propionibacteria acnes. And so look at these two columns here, and what you see is these ones in yellow, we almost only find them where there's acne. 99% of the time, 1% when it's healthy, 99 1, 100 0, and so forth. So this looks like it's that these particular bacteria look like they're bacteria that are contributing to acne or causing it or something like that. They have a pathological effect. But here's one bacteria that is only found in healthy skin. So maybe it's protective. And this is what comes out of microbiome research. Now we have an organism that we could test as a treatment for acne. Maybe what it does, there's a lot of ecological warfare going on your skin. Maybe this is a bacteria that makes toxins or releases other products that kill the other bacteria, and it can therefore take over that part of the skin, but it itself doesn't cause any problem to your skin. So if you added that to a place where you had acne, maybe it would kill all the other bacteria and the condition would go away. So that's just a thought. But okay, diagnosing disease. So this is the neonatal intensive care unit, the NICU, where premature babies live for a number of weeks, 7 weeks, 8 weeks, however long it is, depending on how premature they are before they're released. Here's a little baby. Each of the babies get this very intensive care. There's the baby, and of course they're susceptible to lots of things: infections, necrotizing enterocolitis, respiratory syndrome, lots of things because they're undeveloped, they don't have a microbiome, they don't have much of an immune system. And so this is one area where we do a lot of our work. We in one NICU, we got for about 600 days all the diapers. So for all the babies that were there, we got all their stool samples. And so if something happened to that baby, we could go back and analyze the gut before it happened and look at other babies around that baby to see, how do their guts look compared to that one. So one of the things we see if we look at gestational age is that there's a particular pattern by which bacteria come and go as that baby develops. So this is one of the things you have to do for diagnosis, right? You have to define what's normal so you can recognize what abnormal is. Now, for necrotizing enterocolitis, when we look at that pattern and compare controls, the healthy babies, which is green, to the red, which are the babies with neck, we can see that here's one, this is day of life from birth, and you really don't see it in these cases until these last two groups. So here's one bacteria that is dominant in the babies with neck, but not in the healthy babies. And here's other bacteria that are present in healthy babies and they're absent in the babies with neck. So, number one, we have an opportunity for diagnostics here by looking at these classes of bacteria and seeing if we could see babies that are on the path to the neck before it happens. But now if you think about therapy, perhaps by introducing these bacteria into neck babies and trying to figure out some way of reducing these bacteria, there might be a treatment. This is just a horrible condition, and it's often fatal for the babies as well. Let's see. This is another example of looking at bacteremias in the babies. So these are 7 babies who got bacteremias. This is the day that the bacteremia was identified, day 21. And we can see where it's red, it means that we see the exact same bacteria that we found in the blood in their stool. So this baby was carrying that bacteria 10 days ahead of time before the blood infection happened. And again, if we were to monitor in real time what was going on with these babies, then you might be able to anticipate and intervene before a bad thing happened. Let me just say a little bit at the end here about technology. So I mentioned we sequenced the 16S gene. Illumina is sort of the state of the art for doing this kind of data production, and it's complicated to do. You have to make a PCR product, sequence both ends, and 300 base long Illumina reads are really pushing the instrument to the most it can do. So we're sort of right on the edge of technology to do things as completely as we want to do. We use another sequencing instrument, PacBio, which can do much longer reads, and here we can do a full-length 16S sequencing. So here's a run, and you can see almost all the reads are the full length of 16S sequencing, and we can get a very high accuracy from that. That gives us much more specificity in defining the organism and what's there. And this is an evolving technology. This is a press release from yesterday where PacBio is now announcing a new version of its sequencing instrument. The cost of sequencing will go down tenfold. The amount of sequences you get will go up tenfold. The instrument will be smaller. So sequencing is not done yet. It's still moving along. And here's a couple of postdocs holding a sequencing instrument in their hands from Oxford Nanopore, which we also have started to use for doing DNA sequencing. And I happen to have one in my pocket. So if you don't believe it, here is the first time I've ever given a talk and showed you the sequencing instrument that we use. This works. You will all probably be able to, this costs $1,000 to do sequencing. That's what it looks like. You squirt the sample in, it goes through, you plug this into the USB port of your computer, and the data goes into your computer and gets analyzed. So sequencing technology is still got a long ways to go, and so we will, for microbiome analysis as well as other things, continue to see big advances in terms of our ability. Think of it as a new microscope. We're seeing more and more into the microbial world, and with the new sequencing technologies, it's even going to be more powerful. So let me just wrap up there and put the list of all the people who have done all this fabulous work in my lab. Thank you.
Well, thank you very much for a nice presentation.